All right, we'll start now and hope that the others join the class as soon as possible. I hope that you have finished your last lecture, and by now all of you should be able to join the class. Today is going to be our last lecture for the semester. As I told you the last time, we started with lipid metabolism, looking at lipid catabolism. Today we continue with lipid metabolism, and particularly looking at lipid anabolism today. So that after today, I will send you the last self-assessment questions, which you will use together with the lecture videos, the slides, the book chapters I gave you, everything which you have. From now on, you should seriously use those things to prepare for your exams. All right, so one of the major anabolic pathways of lipids is what we refer to as ketogenesis, which is the synthesis of ketone bodies. Now, we saw much earlier the production of acetyl-CoA from the different pathways, that is from the glycolytic pathway. We saw glucose was producing acetyl-CoA, and from acetyl-CoA that went into the TCA cycle. And that acids as looking at deamination. that I don't know why the microphone is not stable. I hope beginning acetyl-CoA, 2 molecules of acetyl-CoA get conjugated to form acetoacetyl-CoA. And that further goes into the formation of what we refer to as the beta-hydroxy, beta-methyl glutarate-CoA. But what you should pay attention to is that this, you say it's not speaking, really, at all. You can't hear anything. That's strange because the microphone is on. So even as I'm testing it, I can see clearly that everything is working. So I don't know what the issue is. Yeah. Anyway. Can you hear me now? Okay, maybe let me check. Perhaps is the volume of the microphone maybe. Is it clearer now? I've increased the volume to the maximum. Is it clearer now? All right. Somebody says a bit okay. It is. I'm now. I've increased it to the maximum. So I hope that it works now for you, so that you can hear me very clearly. All right. So I think we can continue. Well, we haven't gone so far. So once I've seen this and I attend to it, I hope that it's okay from now till the end. All right, so we're looking at the process of ketogenesis. And I was saying that the process of ketogenesis is that of, acetyl-CoA going into the synthesis of the ketone bodies. Now, the ketone bodies are what, are shaded in the orange colors. So we have acetoacetate, that's a ketone body. And we have acetone, and we have beta hydroxybutyrate. So these three are the ketone bodies, and they are produced from, 2 molecules of acetyl-CoA, undergoing several reactions until you get the formation of these ketone bodies. Now, what you notice is that you see when you have glucose breaking down into acetyl-CoA to go into the TCA cycle. I mean, what that tells you is that acetyl-CoA is an intermediate of the metabolism of glucose. And so if you are having fatty acids also producing acetyl-CoA, what that should tell you is that if you have the condition of diabetes where you have blood glucose in elevated amounts, that will result into acetyl-CoA in elevated amounts. And if you have acetyl-CoA in elevated amounts, then you have some of these acetyl-CoA conjugating to produce these ketone bodies, which is why some of you who perhaps when you went for your industrial attachments, some of you did your industrial attachments in some clinical settings. And perhaps you would have seen that when the blood tests which are done for uh, diabetes, apart from checking for uh, blood glucose levels, there is also the uh, analysis for ketone bodies. So sometimes you see something like hydroxybutyrate on those lab reports. I mean, that is to check to see if, because in conditions of diabetes, you also have the increase of these ketone bodies. If these ketone bodies are found in elevated amounts, I mean, in the system, generally that's referred to as ketosis. You can have that when it's referred to in blood as ketonemia, and if uh, it's elevated amounts in urine, because it can also get excreted in urine, as we saw earlier, that is referred to as ketonuria. Together, this is referred to as ketoacidosis. So, I mean, these words are used interchangeably. The ketosis, ketoacidosis are used interchangeably. But when you say ketonemia, it specifically refers to ketone bodies in elevated amounts in the blood. And ketonuria is referring to ketone bodies in elevated amounts in urine. Now, these ketone bodies can also get broken to produce energy. As we saw, the ketone bodies from the bloodstream move to the extrahepatic tissues, which is the muscle tissues, where they are used for energy. I mean, one of the things that you realize is that, I think I mentioned it before, that the human body is that so strong that uh, it doesn't just give up because of hunger, to say that maybe there isn't enough of, say, the primary source of energy, which is carbohydrate, for it, and so it dies. No, if the body uses all the primary source of energy, which is carbohydrates, everything is finished. There's no carbohydrates again present in the bloodstream. The body will fall on other biomolecules, like the lipids, to generate energy, which is why in starvation, for instance, you have this process of ketolysis, so that when there is a production of this, when you have these ketone bodies that are present in the bloodstream, the body will fall on these ketone bodies, begin to break down these hydroxybutyrate back to acetyl-CoA, which can then go into the TCA cycle to generate energy. So that's what you see there. The ketolysis is the breakdown of ketone bodies. Particularly, we're looking at hydroxybutyrate, which first gets oxidized to acetoacetate, and that acetoacetate further gets oxidized to acetoacetyl-CoA, and then you have the last reaction there, where you have the production of two molecules of acetyl-CoA. And acetyl-CoA goes into the TCA cycle and then generates energy. Now, this process is controlled, is regulated by insulin and glucagon. Insulin is responsible for the regulation of blood glucose, such that it keeps blood glucose levels down, whilst glucagon does the opposite. In other words, insulin facilitates the process of glycogenesis, so that if you have some excess of glucose in blood, they then go into the synthesis of glycogen that will get stored in the liver, or they get stored in the skeletal muscles. On contrary to that, you have glucagon that is responsible for facilitating the process of glycogenolysis, so that when energy is needed, glucagon rather will stimulate the breakdown of glycogen to produce glucose into the bloodstream, which will generate energy. So you then will see that acetyl-CoA, which is, that um, say a final product of that glycolytic pathway, which goes into the TCA cycle. If it is high in the bloodstream, then what will happen is insulin will rather be stimulating the process of ketogenesis, so that there will be lower amounts of acetyl-CoA in the bloodstream, whilst glucagon will be doing the opposite, rather facilitating the breakdown, that is the process of ketolysis, so that you have more acetyl-CoA that can be go into the process of the TCA cycle to generate energy. So these two hormones is not only controlling just glucose levels, as you have known, but even in this process of ketogenesis, ketolysis, you find them also regulating these two processes as well. Now, we go ahead to look at the biosynthesis of fatty acids. We saw the fatty acids to be, the basic units of lipids. And we saw the storage form of these lipids to be the triacyl glycerols, that you have fatty acids that are esterified on the glycerol backbone. And so you have that as a storage form of these lipids. So if you saw the breakdown of fatty acids to generate energy, now we are coming to see the synthesis of it, the anabolism of this. Now, the biosynthesis of fatty acids, we refer to this as de novo synthesis. What it means is that we're having synthesis of these fatty acids from smaller molecules, small, biomolecules. I say small chemical, molecules in the biological system that get conjugated, come together to form these fatty acids. This occurs primarily in the liver, the kidneys. You have this also occurring in the adipose tissue. That is where you have the storage of triacyl glycerol. You also have this occurring in the mammary glands. In the lactating mothers, you have this process of biosynthesis of fatty acids, because milk contains some amount of fatty acids, or say triacyl glycerols. Now, the synthesis of fatty acids require acetyl-CoA, because of course, if you go back to the process of the breakdown, which was the beta oxidation, that major process, you realize that the fatty acid was being broken down in the process of a sequential removal of acetyl-CoA. So you have acetyl-CoA being removed from, say, a long chain fatty acid, one after the other. That's what we saw with that process of beta oxidation. And so if we want to produce fatty acids, then it's going to be a reverse of that process. So we need acetyl-CoA to be joining together to produce the fatty acids. Now, you also have that, you know that the fatty acid is a long, hydrocarbon chain with a carboxylic acid group. And so, of course, the acetyl-CoA becomes the carbon source, the source of carbon, which will give you that chain of the fatty acid. But you also know that acetyl-CoA is a keto acid. What it means is that if you take the structure of acetyl-CoA, it has a ketone group and a carboxylic group, which is to say that it's having some oxygen molecules in there. It's oxidized. But you see the fatty acid is a long hydrocarbon chain. It's just carbon, hydrogen, carbon, hydrogen, carbon, hydrogen until the end before you see a carboxyl group, which means if you have acetyl-CoA, that acetyl-CoA should be undergoing the process of reduction. You should get those oxygen molecules should be removed from the acetyl-CoA. And so you need what we refer to as the reducing equivalent, which is NADPH. And you also need some amount of energy in that process. And that energy is in the form of ATP, which we have seen in various biochemical pathways already. Now, we can break down the biosynthesis of fatty acids into 3 stages. The first stage is going to be the production of acetyl-CoA and NADPH. I mean, this one we have seen it already. We are just going to go over it for you to bring it all together, because we've seen the processes in which acetyl-CoA is produced, and we've also seen the process in which NADPH is produced. We've seen it already, but to go over it and see that, okay, we are bringing everything together, all these metabolism of carbohydrates, metabolism of proteins, and now metabolism of lipids, all of them are coming together. Now, we then look at the conversion of the acetyl-CoA to what we call malonyl-CoA, because it's actually malonyl-CoA that will then go into the synthesis of the fatty acids. And then, of course, we will take a look at a special case of having the same enzyme complex catalyzing a reaction pathway from the beginning to the end. We haven't seen that yet. So far, the metabolic pathways that we have studied, we have seen that each reaction was being catalyzed by a specific enzyme. And so in one reaction pathway, you see different enzymes from the beginning all the way to the end. But in this case, you see that in the biosynthesis of the fatty acids, you have one enzyme complex. So pay attention, it is not an enzyme, but an enzyme complex. So it's you have different enzymes that are in a complex together. And this complex catalyzes the reactions from the beginning to the end. And that is what we refer to as the fatty acid synthase complex. We'll take a look at those reactions that result into the fatty acid biosynthesis. So let's take stage 1, which is production of acetyl-CoA and NADPH, which is something, as I said, we have seen already. We just go over it quickly. Now, we've seen in the process of glycolysis that glucose goes into the production of pyruvate. And this pyruvate that is in the cytosol goes into the mitochondrion. And in the mitochondrion, that pyruvate undergoes a process of decarboxylation. And you have acetyl-CoA produced from pyruvate. Of course, the acetyl-CoA is also produced from amino acids and fatty acids. So you see, where acetyl-CoA we have seen already where they were coming from. They were coming from all these biomolecules. Glucose as a primary source of energy. Then amino acids. After the process of transamination, deamination, the carbon chain goes into the production of acetyl-CoA. Fatty acids, particularly in the process of beta oxidation, also go into the production of acetyl-CoA. And in that, process of um, the TCA cycle, you have pyruvates combining with or conjugating with oxaloacetate to produce citrates. And from the mitochondrion, the citrates can come out into the cytosol back, and in the cytosol, citrates can also go through another process of the production of fatty acids, and these citrates can even go back into the synthesis of pyruvates. So there will be that cycle. But of course, our focus is on where the acetyl-CoA is being produced, and we have seen much of it there is produced in the mitochondrion from glucose, from amino acids, from fatty acids themselves. So it's like a cycle. It's like a fatty acid has produced acetyl-CoA, and that acetyl-CoA then goes back into synthesis of fatty acids again. So that cycle continues. We also saw the pathway, what we refer to as the pentose phosphate pathway. The pathway in which the ribose 5-phosphate was being produced, which went into the production of nucleotides and thus into the production of nucleic acids. That pathway, which we also refer to as the hexose monophosphate shunt, in that pathway, we saw the production of NADPH. So we have seen where NADPH is produced. Right. So now we bring this together and see that we have acetyl-CoA, NADPH, present in the body being produced from these different pathways. Now let's go on to look at the process of the conversion of acetyl-CoA to malonyl-CoA, which is actually a reaction we have seen before. I think when we were looking at the reactions that are catalyzed by the different classes of enzymes. I think this was one of the reactions that we looked at when we were looking at some coenzymes. We saw this coenzyme biotin, one of these vitamin Bs, which is responsible for transfer of carboxyl group onto a molecule. And so we see acetyl-CoA blue at the beginning. It's just one reaction where we see a carboxyl group CO2. ATP is consumed in that reaction there. And we see the enzyme responsible for that reaction, acetyl-CoA carboxylase, because it's a carboxylation reaction. And what you see is just a transfer of that CO2 onto acetyl-CoA to form malonyl-CoA. So it's just a single reaction. That's a transfer of a CO2 onto acetyl-CoA to form malonyl-CoA. So now we have our malonyl-CoA. So we now have the ingredients that we need for the production of the fatty acid. We saw one of the ingredients, acetyl-CoA, which is now getting converted to malonyl-CoA. And we also needed NADPH, which are the reducing agents. We have seen the pathways in which NADPH is produced. We've seen that pathway, the pentose phosphate pathway or the hexose monophosphate shunt. So we have these things. The energy which is needed, ATP, we saw ATP being produced in TCA cycle, in glycolytic pathway. We've seen it. So those things are there already for us. Now, before we go on to the third stage, into the synthesis of the fatty acid, let's take a look at the fatty acid synthase complex, the enzyme complex that I talked about. That in this process of the fatty acid synthesis, you realize that you have one enzyme complex which is catalyzing the reactions from the first to the last, which is a special, thing that we are meeting. It's not like the metabolic pathways which we have seen already, in which you saw that for every reaction there was an enzyme that was catalyzing it. But in this case, you have one enzyme complex that is doing that from the beginning to the end. Now, what you see in this enzyme complex is that see the dashed horizontal line is dividing the enzyme complex into its monomers. I mean, what it means is that the enzyme complex is a dimer. So if you look at the top and the down, they are the same. So it's a dimer, but this dimer is also anti-parallel because you can see the end of the complex below at the right-hand side is the end of the complex above on the left-hand side, and vice versa. Thioesterase, which ends the complex at the right-hand side for the top the top monomer, is now what you see here at the left side for the down monomer. So first thing, the enzyme complex is a dimer. Now, in each of these monomers, you see that there are different enzymes that make the complex. You see the ketoacyl synthase, acetyl transacylase, malonyl transacylase, dehydratase, enol reductase, ketoacyl reductase, and then you have an acyl carrier protein there before a thioesterase. It's the same thing which you find also in the monomer which is below. So you have this subdivision of these two monomers. Now, another thing you should pay attention to is that on this ACP, that is the acyl carrier protein, this is where the synthesis of the fatty acid is going to take place. What it means is that the enzyme complex can catalyze the synthesis of two fatty acids at a time because it's a dimer. And so you are seeing two acyl carrier proteins in that dimer. So simultaneously, you can have the synthesis of fatty acids on both of these acyl carrier proteins. All right. So now what we'll do is we will go into the synthesis of the fatty acids, taking a look at what happens in that process with this enzyme complex. All right, so from what you do is, yes, this is fatty acid synthesis, which is occurring. You look at this as it's a six major reactions over here, 6 major reactions that I mean, what you should pay attention to is that clearly you have this acetyl-CoA, which is blue, and you have malonyl-CoA, which is red, so that we can trace where each of these molecules are going to, the part that they form. And we see the fatty acid synthase is what we have simplified because we have seen it to be a dimer, composed of various enzymes. So that's an enzyme complex. All right. I realized I went offline for a few seconds, so I stopped talking. Now I think I'm back. All right, so as I was saying, we have the fatty acid synthase here, sort of represented in a very simple form. But of course, this is showing just a monomer. It's not a dimer, just to show one part of that complex, which means here you are seeing the synthesis of one fatty acid, but not two at a time. In the first reaction, what you see is acetyl-CoA comes to bind on the acyl carrier protein. So we see that. First, you see the coenzyme A is broken off from the acetyl group. And so what you see is you have the acetyl group binding on the acyl carrier protein. The next thing you see is that acetyl group, which is bound on the acyl carrier protein, moves to the end of the enzyme complex. I mean, remember that enzyme complex is consisting of, a large number of enzymes there. And so it moves to the last end where you now have that acyl carrier protein free to receive malonyl-CoA. So now malonyl-CoA comes to bind onto that acyl carrier protein. What you see is that after the second reaction, you see both the acetyl group on that end of the enzyme complex and the malonyl-CoA on the acyl carrier protein. Both of them are now bound on the enzyme complex. Then the next is you see that carboxyl group, which is on malonyl-CoA, is cleaved off. So there is that decarboxylation in that malonyl-CoA residue. Now you have the acetyl group from that end now coming to join onto the malonyl-CoA residue. So now you see the blue and red parts joining together. So you now have four carbons that are there on the enzyme complex. Now, what happens next is some, reduction reactions, because now you see NADPH comes into the reaction, and you realize that those ketone groups, so the first ketone group, this blue group, the C double bond O becomes OH. The next thing you see is that OH is removed. There's now a double bond because there is some hydration reaction in that dehydration reaction. That is reaction number 5. The reaction number 6, NADPH comes again. That is another reduction process where now you have that double bond, which was between the malonyl-CoA and the acetyl-CoA residues removed. And you see there are now 2 hydrogens on those carbons. Now, in the end, what you should come to realize is that when you take acetyl-CoA, which contains 2 carbons, and you take malonyl-CoA, which contains 3 carbons, but 1 carbon is removed in a decarboxylation reaction for malonyl-CoA, you are left with 4 carbons. So 4 carbons are now joined on the enzyme complex, 4 carbons. But in order for you to have a fatty acid, a fatty acid is a long chain fatty acid. And usually this process of fatty acid synthesis ends with the production of palmitic acid, which contains 16 carbons. So if you have 4 carbons that are produced in that first reaction, and the process is such that you have an addition of 2 carbons at a time. And that carbon that is being added on and on, is coming from malonyl CoA. Then the reaction number 2 to number 6, when it is repeated, so that reaction repeats itself from reaction number 2 to reaction number 6, which is addition of 2 carbons, 2 carbons, 2 carbons, 6 times. That will become 12 carbons. Just like we saw with the addition of the breakdown of 2 carbons at a time in the process of beta oxidation. This time around, what you are seeing is that there is addition of 2 carbons at a time, 2 carbons at a time. But that addition of 2 carbons is occurring from reaction number 2 to reaction number 6 in order for us to get the full palmitic acid molecule, which is consisting of 16 carbons, which is palmitate. Now, we have palmitic acid, which is produced in this process of the fatty acid synthesis. But we know that that is not the only fatty acid that we have. We have other fatty acids, which are, consisting of more carbons or less carbons. We even have fatty acids that when we did a classification of fatty acids, we saw saturated and unsaturated fatty acids. And palmitic acid, as we can see, is a saturated fatty acid. So how then do we get these other fatty acids after we've gotten the palmitic acid? How do we get this? So for the unsaturated fatty acids, we get them by the process of desaturation. So in this process, you have also specific enzymes, desaturases, which are responsible for the removal of hydrogens from the carbon chain, which will result into the formation of double bonds. So you can say it's a dehydrogenation reaction. So the palmitic acid, which was produced, consisting of 16 carbons. What is being shown here is that, first of all, you have a process which is referred to as elongation, but we'll come to that process separately, elongation. So first, you have a 16 carbon palmitic acid, which is converted to steric acid because you have an addition of 2 carbons to it again, so that you now have 18 carbons before you start having a process of desaturation. Now you see the enzymes are being referred to with the carbons on which the double bonds are formed. So for instance, if you see delta 9 desaturase, what it means is that the double bond which is formed is on carbon number 9. In other words, that double bond is going to be between carbon number 9 and carbon number 10. That is where the desaturase is acting directly. You also have delta 12 desaturase, which is responsible for the formation of a double bond between carbon number 12 and carbon number 13. You have that similarly also for delta 15 desaturase. So the enzymes that are responsible for the formation of unsaturated fatty acids are desaturases. And specifically, if you want to refer to the names, you can use the position of the double bonds to specify where those enzymes are acting. I think because we did the structure of the fatty acids, the various ways we present them. I think it should be easy for you to relate to these things. Delta 9, delta 12, delta 15. We have done those things already, so you can easily relate to the formation of those double bonds, the position of them in those unsaturated fatty acids. Right, now we can look at the process of elongation. Now we can look at the process of elongation. Now the process of elongation occurs in two places, either in the endoplasmic reticulum, what you're referring to as the microsomal chain elongation, which is the addition of malonyl-CoA by elongases. So take note that this time around, the addition of malonyl-CoA is not being done by pay attention that this time around, the addition of the malonyl-CoA for the chain to get long is not being done by the enzyme complex that was involved in the process of the fatty acid synthesis from the beginning to the formation of the palmitic acid. But this time around, this is being done by different enzymes, what we are referring to as elongases. You can also have the fatty acid elongation occurring in the mitochondrial, in the mitochondrion, what we refer to as the mitochondrial chain elongation. This one is almost like the reverse of beta oxidation, because you see the beta oxidation was a sequential removal of acetyl-CoA. So if now you are having an addition of acetyl-CoA, two carbons in order for the chain to get long, this occurring in the mitochondrion, then that becomes a reverse of beta oxidation. So you see the two differences. You see the differences between the macrosomal and the mitochondrial chain elongation. In the macrosomal chain elongation, you have malonyl-CoA, which is being added. But in the mitochondrial chain elongation, you have acetyl-CoA, which is being added in order for the fatty acid chain to get longer. Now, you do have that, after you have synthesized these fatty acids, which we saw as, the I think our network has not been so good today, but anyway, anytime it breaks, I just stop talking and wait for some time so that we don't miss anything. What I was saying was that you have fatty acids as one of the building blocks of lipids. I mean, in addition to glycerol as a backbone, you have now synthesized fatty acids. But then you do go into the synthesis of triacylglycerols, which are the storage forms of these fatty acids, particularly the storage that goes into the adipose tissue. And we see that occurring from, the glycerol backbone, from the top, glycerol backbone. The first thing you see is that there is a phosphorylation of that glycerol molecule, which is one of these common things we have been seeing from the beginning, that molecules get phosphorylated as a form of activation of these molecules to go down a particular reaction pathway. And so you have phosphorylation of glycerol to have glycerol 3-phosphates. Now, that glycerol 3-phosphates can serve as a parent molecule for the synthesis of even phospholipids, because now there's a phosphate group which is attached on that glycerol. So that when you have the first fatty acid, which is getting esterified on that carbon, of course, that's coming from some acyl CoA. And then you have a second acyl CoA, which is also coming, so that you now have a second fatty acid bound on the second carbon. You now have phosphatidic acid. So actually, it is this phosphatidic acid from this phosphatidic acid that you refer to it as the parent molecule for the synthesis of phospholipids. Because from it, any modification to this molecule will result into the production of different kinds of phospholipids, which we have seen as a major component of cell membranes. We also see that glucose, in the glycolytic pathway, we saw along the line there was a production of dihydroxyacetone phosphates. And so if you have dihydroxyacetone phosphates, these can go into the synthesis of the glycerol 3-phosphate and go down that pathway. It's just by the reduction of that ketone group at the center there to a hydroxy group, you have a glycerol 3-phosphate. I mean, we've seen this reaction. We've seen this conversion somewhere along the glycolytic pathway. Or you can have that dihydroxyacetone phosphate now getting conjugated to a first fatty acid molecule to now have an acyl dihydroxyl acetone phosphate. And that too can go into the formation of lysophosphatidic acid by the reduction of the ketone group at that central position of the molecule to now a hydroxyl group. This comes back to the formation of phosphatidic acid as a parent molecule for the synthesis of phospholipids. You can have that phosphate group, which is on phosphatidic acid, hydrolyzed by phosphatase. So you now have a diacylglycerol. And I think we have seen diacylglycerol in some of these glycolytic pathways. Okay, maybe not you. There was one of these groups I was taking that they were doing some things on I think cell communication, and we are referring to the function of hormones, and you see diacylglycerol as a signaling molecule acting in cell recognition there. But from here, from the diacylglycerol, just by the addition of one fatty acid on the third carbon of the glycerol backbone, you have the synthesis of triacylglycerol. So from glycerol and then the addition of the fatty acids throughout all these pathways, you have the synthesis of triacylglycerol at the end, which is the storage form of fatty acids. Mainly, you find that storage in the adipose tissues under the skin. All right. Then, of course, you can have the synthesis of phospholipids also from this, where you have phosphatidic acid from that pathway. Either you have the phosphate group hydrolyzed off to form a diacylglycerol, or you can even have further addition of even a nucleotide like cytidine, which gives you even a further molecule for signal transduction, for cell communication. From this, you can have the synthesis of various molecules like phosphatidylinositol, cardiolipin, phosphatidylethanolamin, phosphatidylserine. So you see, this is an interesting one that you see phosphatidylethanolamine, phosphatidylserine. This is telling you are now having a complex of lipids with amino acids. So you see how you are having the interconnection of the metabolism of these biomolecules. And all these molecules like phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine, they all act as some receptors on the surface of the cell responsible for cell communication. So they are embedded in the cell membrane. All right. Now we can also take a look at the synthesis of cholesterol, which is not a straight chain molecule, but that is a structure with some, rings. We have a ring structure for cholesterol. And in the beginning, we see the synthesis of the cholesterol precursors. The cholesterol precursors, where here too, we see that this begins with two molecules of acetyl-CoA forming acetoacetyl-CoA, which goes into that formation of the final molecule there, mevalonate, or hydroxymalonyl glutaric-CoA, which goes into this ketone body synthesis, this thing we have seen much earlier. Now, what we also see is that from here, you have these two are the precursors. What we are seeing, the isopentenyl pyrophosphates and dimethyl allyl pyrophosphates, these two are precursors. It is these two molecules that go into the synthesis of cholesterol. We do have some drugs for lowering cholesterol levels in blood, and they actually target this enzyme, the HMG-CoA reductase, so that its inhibition will result into a reduction of the production of these cholesterol precursors. Now we have the precursors, the dimethyl allyl pyrophosphates and the isopentenyl pyrophosphates. These two precursors conjugates. So it's just, if you see the dimethyl allyl pyrophosphate, it's red with two phosphate groups on it. And you have the isopentenyl pyrophosphate, which is blue with two phosphate groups on it. So you can easily see the conjugation, which means two phosphate groups are removed from the isopentenyl pyrophosphate to have a conjugation with it, with a dimethyl pyrophosphate. That gives you geranyl pyrophosphate. And you have two of those that get conjugated to produce squalene. And squalene then goes into the synthesis of lanosterol. So actually, lanosterol becomes that very first cholesterol nucleus which is produced. So this structure, you see it in cholesterol derived molecules. A six-sided ring, three six-sided rings fused together this way with a five-sided ring. You can have different side chains on them, and all that will give you the structure of the steroid nucleus, what we refer to as the steroid nucleus. Now, from lanosterol, you then will have the production of cholesterol. And from cholesterol, you have the process of steroid agenesis, from which you have the production of different forms of steroids, and these are different hormones like the uh, progestagens, pregnenolone, progesterone. I mean, these are some sex hormones or the precursors to the synthesis of female sex hormones like the estradiol, estriol, what we refer to as estrogens. You also do have the production of the androgens. You mainly talk about testosterone, dihydroxytestosterone. All of these are androgens. Then you also do have some of these adrenal hormones, mineralocorticoids, responsible for the regulation of minerals and water balance in the body. So aldosterone, corticosterone, cortisol, being that hormone for regulating stress, response to stress, deoxycorticosterone, all of this. If you just pay attention to just the ring, just take a look at the ring, you see all of them are having the same ring, just like cholesterol. Six sided ring fused together with one five-sided ring. I mean, if you don't understand anything at all, if you see the steroid nucleus, the structure of the steroid, you should quickly know this is a steroid, because you will see the rings are the same. Six-sided rings, three of them fused together with one five-sided ring there. That's what we refer to as a steroid nucleus, and that you find in all steroid hormones. Now you can also pay attention to the color coding, such that these colors, say these orange colors, refer to the processes that are occurring in the mitochondrion, and then or say those enzymes are in the mitochondrion, and the green, you find them in the endoplasmic reticulum. So those processes too are going to be microsomal. All right. We also do have what we refer to as a sphingosine. I think we did look at some membrane lipids at the end of our last lecture. We did look at these membrane lipids, and these membrane lipids do have the sphingosine as a backbone. So in place of the glycerol as a backbone for triacylglycerols, you find sphingosine, which is a backbone on which other fatty acids are bound onto, or you have some even oligosaccharides bound onto this sphingosine, which gives you the structure of membrane lipids, particularly in the nervous system. You have these membrane lipids, which give some protection to the neurons. Now you see the synthesis of the sphingosine is coming from serine and palmitoyl-CoA. So that tells you an amino acid, serine, is involved in the synthesis there with the palmitic acid, which is produced in that process of the fatty acid synthesis that we saw. And through several reactions, you have the production. And through several reactions, you have the production of the sphingosine. Of course, you can see along the line, you can have that 3-keto sphingosine going into the production of dihydrosphingosine. But our focus is on this, the sphingosine, which you find in these membrane lipids. And so we see, I mean, here, I think we will let's take a look at the different types of sphingolipids that you can come across, particularly in the the nerve cells. So this is a sphingosine. The first you are seeing there is a sphingosine. If you pay attention to it, you realize that that is just palmitic acid with hydroxyl group. On carbon 1, carbon 3, and on carbon 2, you have an amine group there. Where would that amine group come from? Definitely from serine, because we just saw the synthesis is starting with serine and palmitic acid. And so on the amine group, you can have some acyl group, a fatty acid, which is bound on there. That will give you some sphingolipid, so ceramide. You can also have, apart from an acyl group binding on the amine group, you have some phosphocholine, you have a sphingomyelin. I think these two, the sphingomyelin here, and then you have another one, phosphoethanolamine, also bound on that first carbon there. That gives you also another sphingomyelin. These two should ring a bell because in secondary school, you would have learned the structure of the neuron, where you drew the structure of the nerve cell, having the cell body with dendrites, and then you had this axon, and along the axons you had a protection, what you refer to as a myelin sheath. So that quickly should tell you that you have these sphingomyelins as major component in those myelin sheaths, giving protection to the stem of the nerve cell. You can even have carbohydrates. So you see, that's a sugar, like a glucose, that is bound on that first carbon there. That gives you a cerebroside. I mean, the name cerebroside should tell you that this definitely is in the nervous system, because you refer to cerebrum in the brain, you refer to cerebrum in that central nervous system. So if you see cerebroside quickly, you also will see ganglioside. You refer to the ganglion also in the central nervous system. And here you see that's a more complex structure there. You have some sialic acid and some oligosaccharide there. So you see that sphingolipid is consisting of the sphingosine. You have some phosphocholine. You have phosphoethanolamine. You have a monosaccharide. You have also some oligosaccharide. All these are forming complexes to give you these sphingolipids, which are major components of the cell membranes of these neurons. And I think this is where we ended the last time. And so now you can relate to that you have this sphingolipid, which consists of, that sphingosine and some oligosaccharides. And these oligosaccharides, we saw this, this color coding. The blue, the deep blue means glucose. The yellow means galactose. The white means N-acetyl-galactosamine. The light blue means 5-acetylnuraminic acid. And so we talk of the fact that you have these membrane lipids that undergo some recycling, and the process of recycling they undergo is catalyzed by various enzymes, such that if you have the deficiency of these enzymes, you have the accumulation of some of these sphingolipids, which results into some neurodegenerative disorders like the generalized gangliosidosis, which is due to accumulation of GM1 because of the deficiency of beta galactosidase, so that there will not be the breakdown of GM1 to GM2. Similar thing, if you have the accumulation of GM2 because of the deficiency in hexosaminidase A, you have the condition of the Tay-Sachs disease. You have the glucocerebrosidase also, resulting from the Gaucher's disease, resulting from the deficiency of glucocerebrosidase. Deficiency of hexosaminidase A and B will result into Sandhoff's disease because you have an accumulation of this globocyte. The Fabry's disease results from a deficiency of alpha galactosidase A. The Niemann-Pick disease is resulting from deficiency of sphingomyelinase. We referred to these things in our previous lecture. So this is just to bring it back for you to relate it to, the structure of the sphingolipids, which we have just looked at. So where we ended the last time, that is where we are ending again today. All right. So thank you so much for your audience. The floor is open now. If you have any questions, if you have any contribution, any comments you would like to make, you can write in the chat, or you raise your hand. I will enable your microphone. You unmute yourself and you speak. The floor is open.